Welcome to the module, Steps of a Systematic Review. In this module, I will describe how to conduct a systematic review. We will go through the first 5 steps, so looking at how to develop a research question, how to write a study protocol, developing a search strategy, performing study selection, and assessing study quality. In the third and final module, we will cover data extraction, meta-analysis, and preparing your results for publication. To begin a systematic review, it is important to define your research question and to write a protocol. The research question of a systematic review should be feasible, interesting, and relevant. Therefore, a clear, logical, and well-defined research question should be formulated before beginning work on the review. When it comes to formulating your research question, it is worth considering the components of the PICO model. PICO is a widely used tool for formulating clinical research questions and search strategies, and we can apply the PICO model to preclinical questions. PICO stands for 4 different potential components of a question. The first, P, is for patient, population, or problem. So here we would consider what is the condition or disease of interest and what are the characteristics of the patient or population. Under I, we have intervention, which is the intervention or exposure under consideration for this patient or population. C is for comparison, so what is the alternative to the intervention? For example, this could be a placebo control, a different drug, or a surgery. And outcome is what are the relevant outcomes. For example, in a clinical situation, we might be interested in the outcome of morbidity, remission, or any complications associated with a treatment. It is worth remembering that not all research questions will fit this format. Other review questions will follow a different question structure, perhaps only using some elements of the PICO. As an example research question, we will consider a preclinical systematic review published in Stroke in 2014. Here, the review looked at the efficacy of antidepressants in animal models of ischemic stroke. If we think about the question structure using PICO, the population they were interested in was animal models of ischemic stroke. The intervention was antidepressants. The comparator group were animals that had received vehicle or no treatment, and the primary outcome measure was infarct volume. It is important to define these elements of your research question from the start to ensure you design an appropriate search strategy to answer your research question. As I mentioned, other review questions will follow a different question structure, perhaps only using some elements of PICO. For example, a recent systematic review used a broad-scoped approach to investigate the current evidence for how the cerebral microvasculature responds to lactate in studies ranging from the cellular level, so in vitro and in vivo studies, to human experiments. The aim was to identify gaps in knowledge which may provide further insight into future experimental hypotheses and novel treatment avenues. There are many different examples of how you can formulate a research question. A review protocol outlines why and how you are going to conduct your systematic review. It describes the rationale, research question, background, hypothesis and methods that will be used, including the search strategy, inclusion criteria, data extraction, quality assessment, data synthesis, and statistical analysis plan. A protocol should be prepared before a review is started and used as a guide to carry out the review. Having a pre-specified protocol improves the methodological transparency of your review and reduces the risk of introducing bias by enabling comparison of reported review methods with what was planned. It also helps to avoid unplanned duplication of reviews. There are guidelines to help write a systematic review protocol. For example, the PRISMA guidelines set out what should be included in a systematic review protocol in detail. For preclinical reviews, we recommend using the CIRCLE protocol format, and this is for systematic reviews of animal intervention studies and are shown here. These guidelines should be used to ensure that you cover all the necessary components in your protocol. Your protocol should be made publicly available. The options are to register, share online, or to publish your protocol. To register, Prospero is an international prospective register of systematic reviews, which accepts both clinical and preclinical systematic reviews. There's also the option of Cochrane, which accepts clinical systematic review protocols only. To make your preclinical protocol public, our group, the Camarades Group, and the Circle Group host preclinical protocols on our websites. Publishing your protocol in a journal, for example in the BMJ Open Science or Systematic Reviews journal, gives you the opportunity to gain feedback on your methods and research question, etc. This process of journal, sending your protocol for a review, and giving you the opportunity to respond to reviewer comments can be very valuable for refining your question and methods. Another option for publication is via the Open Science Framework, where you will receive a DOI for your protocol. The next step is to identify relevant studies, which involves developing and implementing a search strategy. Firstly, select the databases that you will search, for example PubMed, Embase and Web of Science. There's also the option to search the grey literature, such as the reference lists of included studies. The databases you select will depend on your topic and the journal coverage provided. It is generally accepted that you need to search more than one database to guarantee adequate and efficient coverage. These databases and search strings need to be pre-specified in your protocol. The study highlighted here has assessed the retrieval across different databases and found that to reach maximum recall, defined as the number of relevant reports identified divided by the total number of relevant reports in existence, searches should include a combination of databases to guarantee adequate and efficient coverage. As I mentioned, the databases we search will depend on our research question. In preclinical reviews, we would usually search PubMed, Embase, and possibly Web of Science. There is also the option to search Google Scholar and Scopus. Preprint servers may also give you access to relevant data, and it may be appropriate to search bioRxiv or medRxiv depending on your research question. We search more than one database to give us a higher chance of obtaining all relevant papers. The search terms used in your search strategy are based around each of the concepts in your research question. Here, if we take the systematic review question we gave as an example previously, we would search for the population, so animal models, the disease, stroke, and the intervention, antidepressants. We would not usually include the outcome or comparator in the search terms, as these are often not reported in the title and abstract of publications. The overlap between these three domains, so where all three of these are included in a publication, are the publications that we are interested in. For each of your research question components, you should try to consider all relevant alternative terms and synonyms. For example, stroke or cerebral ischemia or cerebral vascular accident. And for the intervention type, here antidepressants, you should also list known antidepressants that you will include in your review. We need to develop a search strategy for each database that we search, and database-specific things to consider are index or subject terms, for example MESH terms and Emtree. The MESH database can be used to look for synonyms to use as free text. It's worth noting that MESH is hierarchical, and stroke is part of cerebral vascular disease, and under stroke we get more specific terms. You can use PubMed to filter for animal studies by using the checkbox, or Circle have published for animal filters for PubMed and Embase. It is important to think carefully about how you link your search terms using Boolean operators. We would use OR in a search to connect two or more similar concepts, for example stroke or cerebral ischemia. This broadens the results by telling the database that at least one of the search terms must be present in the results. We would use AND in a search to narrow the results and tell the database that all search terms must be present in each result. Firstly, if we consider the terms used for the intervention and disease that we are interested in, we can set what fields to look in using the search details field. For example, we can search for these terms in the title and abstract, or the MESH terms by using square brackets after each term and stipulating where you would like to search. We can combine the first and second part of our search with AND. This will identify all studies reporting the intervention and disease that we are interested in. Finally, we can use the PubMed animal filter and retrieve all publications that describe stroke, antidepressants, and using animals. In any search, we are trying to get a balance between precision and sensitivity, where sensitivity is our ability of a search strategy to identify all relevant articles, and precision is our ability of a search strategy to exclude irrelevant articles. We aim to maximise sensitivity while attempting to maximise precision. A sensitive search retrieves most relevant literature, and the risk of missing relevant literature is low. However, the drawback is that it retrieves a higher proportion of irrelevant literature, meaning more time is spent filtering and screening. A precise search retrieves some relevant literature, retrieves fewer irrelevant studies, and less time is spent filtering and screening. However, there is a high risk of missing relevant literature. This is why the balance between sensitivity and precision is so important. Once you have run your search strings, you should import those results into a reference manager software, for example EndNote. After searching across multiple databases, it is important to deduplicate your results. This can be done in EndNote, or the Camarades group have developed a Shiny app using R code to do this. It is important to keep a record of the number of records obtained from each database and the number of duplicate references identified. The next step is to retrieve the full text PDFs using either EndNote or Zotero. It is worth investing time to make sure your search strategy is effective and efficient, as the quality of the search strategy determines how comprehensive your review is. As we've seen, search strategies are database specific, and one option is to use Polyglot to convert search strings to different databases. Information specialists can also be very helpful in the development of the search strategy, and contacting the library could be useful. Finally, there is a PubMed tutorial to help you with this step in PubMed. The next step in the review process is to identify relevant studies, also known as screening for inclusion. Your search may produce a large number of potentially relevant articles, and these need to be assessed for relevance against the pre-specified criteria set out in your protocol. The inclusion and exclusion criteria to be used during screening should be predefined in your protocol and usually set before the search strategy is developed. You should also think about any language restrictions you're going to put in place and what can be translated. You should think about publication date restrictions. This might be important if you're doing a review update. And you should think about the type of publications you'll include. So will these be peer-reviewed? Will you include abstracts? It's important to bear in mind that every time you place a restriction on your review, you're potentially introducing bias. Screening is the stage where we decide whether a study should be included or excluded in your review. It is recommended that 2 independent reviewers screen studies for inclusion, and a third screener can arbitrate if a decision cannot be reached. Screening is often conducted in 2 stages. This might be first stage looking at title and abstract, which uses the title and abstract only to reach a decision. It is important to be over-inclusive at this stage to prevent the incorrect removal of relevant papers. The second stage may take those papers that were included in the first stage, and the full paper is used to make a decision. This relies on a more detailed assessment, and for studies that are excluded, it is useful to keep a record of why they are excluded. The PRISMA flow diagram or similar chart can be useful to visualise and report the study selection process. There are different software options available for helping with the screening stage of your review. These can help you track the inclusion and exclusion decisions. For example, our group have developed a SURF software, which is specially designed for preclinical systematic reviews. There's also EpiReviewer, which is a web application that enables researchers to manage the entire life cycle of a review in a single location. There's Abstracker, which is a free open source screening tool. Covidence, which is the Cochrane version, which is an online systematic review management, including screening, risk of bias assessment and data extraction. And others have used reference manager software, for example EndNote, for the screening stage of their review. Once you have completed the screening stage of your review, the next step is to extract study characteristics and assess study quality, or risks of bias. You will extract relevant data on study characteristics, which you predefined in the review protocol. This can actually be done during the screening process or once you have screened and included studies. The data you extract may include information on PICO, so the population, intervention, comparison and outcome, and this may include details of species and strain of animal, dose and timing of intervention, and the type and time of outcome assessment. We would also take out information on the methodological quality of the study, and in this context, when we consider study quality, we're assessing the methodological and reporting quality, so how the study was done and how well it was reported. Understanding the quality of the evidence that goes into a review is vital to the interpretation of the findings. We introduced the concepts of study bias in the introductory module. Here we will recap on the concept of study bias and how we can assess for these when we are looking at the quality of the papers included in our review. So why are we interested in study bias? Bias is a systematic error introduced into the experiment that leads to distorted results and affects the internal validity of a study, or the confidence that we can place in the cause-effect relationship in a study. Adequate internal validity of an animal experiment implies that the differences observed between groups of animals allocated to different interventions may, apart from random error, be attributed to the treatment under investigation. The internal validity may be threatened by 4 types of bias. These are selection bias, performance bias, detection bias, and attrition bias. For example, in an animal experiment testing a treatment, selection bias is the systematic difference in baseline characteristics in these groups of animals, and this can be addressed by randomization. Performance bias is the systematic differences in the care between treatment groups, apart from the intervention under study, and can be addressed by allocation concealment. Detection bias is the systematic differences between groups in how outcomes are determined when the person assessing outcome has knowledge of treatment assignment, and can be addressed by blinded assessment of outcome. An attrition bias is the unequal occurrence and handling of deviations from protocol and loss to follow-up between treatment groups, which can be addressed by pre-specifying exclusions, reporting dropouts, and intention to treat analysis. So these 4 items, randomization, allocation concealment, blinded assessment, and reporting of dropouts, can all be assessed in the reporting of a paper. A range of tools exist to help assess risks of bias in different study types. Specifically for studies using animals, we have the Circle Risk of Bias tool and the Camrades checklist. The Circle tool is not a yes, no, it was reported or not, but also takes into account the method used and whether the study is at high, low, or unclear risk of bias. It is important to always pre-specify how you will assess risk of bias in your study protocol. There are different data extraction software available. So similar to the screening software, we have SURF, EpiReviewer, and Covidence. There's also SRDR Plus, which is a free open source data extraction and management tool. So it's worth considering which of these are the most suitable for your project before you begin.